Good afternoon, everyone. Today we will discuss how high-intensity exercise can slow down our aging process. In fact, the process is caused by telomere, and a detailed explanation will follow. As a first step in this video, we will give you a brief overview of DNA replication, structure of genetics material, and how they work. In the next session, we will discuss how exercise affects telomere length and its relationship to biological aging. And telomerase will also be discussed within this context. At the end, some limitation of this research and summary will be included. We align our topic with BABS themes 2 and 4 on cell biology, architecture, and genetics respectively. Several learning outcomes are also met, including learning outcomes 1 and 2 on DNA replication and learning outcome 1 on cell division and replication. Okay, so let's start by looking at what are telomeres. There are actually 6 DNA sequences in this region, TDA-GGG. It is located at the end of chromosomes and usually repeat about 3,000 times. Telomeres protect the end of chromosomes from nucleotide degradation, prevent fusion with other chromosomes. However, its most important function is to allow chromosomes to replicate properly during cell division. And a DNA is a double-stranded molecule, which is an anti-parallel double helix that runs from 5' end to 3' end with 4 bases: adenine, thymine, guanine, and cytosine. A will pair with T and C will pair with G, and the nitrogenous bases are held by hydrogen bond. After explaining the structure of DNA, now let's move on to the DNA replication. This process will occur before cell division. During the DNA replication, it is started by a helicase, which unwinds and separates the parental DNA strand and lets the small RNA primer stick onto it, guiding the starting point, and followed by the accurate replication of the genome by a DNA polymerase, which always adds a nucleotide to the hydroxyl group at 3' end and forming the leading strand. It is interesting to know that as the DNA molecules unwind little by little, the RNA polymerase lays down the primers, and the lagging strand is then generated bit by bit, which is called Okazaki fragment. After replication, the primer's RNA is replaced by DNA nucleotides by DNA polymerase 1. The lagging strand, however, does not have a 3' end for DNA polymerase to work on. This means a single strand overhang occurred at the end of the chromosome, and a part of the DNA is not copied in each run of replication.
This is where telomeres come in. They are often described as the protective caps at the end of chromosomes, similar to the plastic tips of a shoelace that serve to protect DNA and are also used for signaling where the DNA starts and ends. As we know, our DNA replication mechanism system isn't able to copy the ends of DNA properly, resulting in the end replication problem. This would be a serious issue if important genetic material was being lost during this process. However, these telomeres act as a buffer and are situated at the end so that they are given up instead. Over time, as your cells keep dividing, your telomeres keep getting shorter and shorter, and this process is known as telomere attrition. So one can say the shorter your telomeres are, the older your cells are. In this way, telomere length can serve as a biomarker for age. When telomeres get too short, the cell initiates the DNA damage response, whereby cells stop dividing and become senescent cells. Cell senescence due to telomere shortening is called replicative senescence. The cell is still active in this state, however it cannot replicate anymore, and this is done to prevent genetic material from being lost in further rounds of replications or any impending DNA damage. Our cells can divide around 60 times before cell senescence occurs, and this is known as the Hayflick limit. From here, our cells can either undergo apoptosis, where the cell is programmed to die and clear off, or restart up again by activating an enzyme called telomerase, which has the ability to replenish or build telomeres, allowing cells to divide again. So, in order to live forever and reverse aging, we just need a lot of telomerase, right? Sadly, this is not the case. The more times a cell has divided, the more likely that it is to have acquired a dangerous mutation along the way, which could lead to cancer, and is why cells die off in the first place, to stop this from happening. In fact, cancer cells divide uncontrollably because they have a large amount of this enzyme. So while increasing telomerase may allow you to live longer, but not necessarily healthier, as high telomerase levels elevates the risk of developing cancers. There is, however, a solution that can help preserve our telomeres and may allow us to live a couple extra years, and that is exercise.
Exercise shares a unique relationship with telomere length. There have been claims that intense physical activity can slow down aging by almost 9 years. However, studies suggest for these benefits to be achieved, exercise must be of high intensity and consistency. Exercise can help lengthen or maintain telomeres by increasing telomere activity. Telomeres are enzymes within the body with the primary role of maintaining telomere length and health. Exercise stimulates telomeres activity, thus aiding with telomere maintenance. Think of telomeres as a repair crew that adds new rope to an end as it frays over time. The more frequent and active the repairs, the longer and healthier the rope. Exercise also reduces oxidative stress and inflammation, which can slow the shortening process of telomeres. Oxidative stress refers to breakdown of cells over time. Just as mold causes a fruit to spoil and become unfit for consumption, oxidative stress damages the cells in your body and leads to various health problems. Essentially, regular exercise can help lower the harmful effects of stress and inflammation in our body cells. On top of this, regular physical activity has also been associated with healthier aging and can help to prevent or reduce the risk of age-related conditions such as heart disease, and may contribute to overall healthier and more independent lifestyle in the later years. In 2017, Tucker conducted a study bringing together 5,800 adults and measuring their telomere length, finding that leukocyte telomere length, LTL, was significantly longer in those who engaged in high-intensity physical activity, with a high-intensity group having on average 140 base pairs longer than sedentary counterparts, concluding that exercise was a key intervention strategy in slowing telomere shortening. In 2015, Borghini brought together 20 ultramarathon runners, averaging a total of 60 kilometers of running a week. Measuring telomere length, he compared the athletes to 42 matched sedentary controls. He found that the endurance athletes had substantially longer telomeres, even after adjusting for cardiovascular risk factors, with the greatest difference being seen in those who were the oldest and been consistently training for the longest. He concluded that to significantly stimulate telomere activity, exercise must be of consistent high intensity. Conversely, in 2015, South and Roth looked at telomere length of 815 elderly patients with varying exercise habits, only finding significant evidence for slow telomere aging in those who had consistently been exercising for over 41 years. Interestingly, a study revealed that those who had exercised for less than 10 years of their life had similar telomere aging for those who had done no exercise at all, revealing the need for exercise to remain consistent to have a benefit. While there are a multitude of studies showing the relationship exercise has with telomere length, it is important to recognize that exercise isn't the sole determinant of telomere length. Age, genetics, hormonal factors, psychological stress, and exposure to carcinogens all influence telomere length and aging.
While telomere length is a legitimate identifier for aging, the effects of exercise on telomere length are still not properly understood. How does exercise reduce the shortening of telomere length is still a valid and unanswered question within this research field. From what researchers have found, there are two possible main reasons to why physical activities reduce shortening of telomere length. One of these reasons is the hormone irisin, which is released in the skeletal muscle and is directly related to telomere length. Irisin has also been researched to progressively increase when a person exercises. Another possibility is oxidative stress. Within studies, oxidative stress has been found to be associated with the acceleration of telomere shortening. And it has been found that when we perform physical activities regularly, oxidative stress has decreased. You may be asking, what is oxidative stress? Well, oxidative stress occurs when there are too many free radicals built up in your body and there are not enough antioxidants to counter the buildup. This causes damages to cells, proteins, and what we previously said, telomeres. Finally, for limitations, many studies do not consider sleep activity and mental well-being such as stress, depression, or anxiety of the patients. Both factors have been widely documented for the correlation to telomere length and the rate at which it shortens. In summary, we have explored what telomeres are and how they are important in every living organism to preserve DNA when replicating. We have also learned that telomere length closely related to aging and how telomerase is commonly found within cancers. Furthermore, the topic of exercise was covered with how it is associated with preserving telomere length within multiple studies. And finally, the limitations to said studies were analyzed and explained. From this, we all hope that you have enjoyed and learned something new about